can get started. So we are particularly fortunate today to have Professor Bernhard Palsson from the Department of Bioengineering at UC San Diego. And that's the title of his talk. I'll tell you a little bit about it. But the point of this introduction is just really to introduce a bit the speaker. And you have to know we are particularly fortunate. I call him Mr. Systems Biology because he more or less started, in my opinion, the best work that was done on systems biology. And there's several aspects to it. And one is just trying to get the metabolic maps. So anybody who works on E. coli, his metabolic map that has 904 of the genes involved in it is just so widely used by people. And there's been so many experiments starting in the 2000 to now, looking at from environmental applications, biotechnology applications. And then, of course, he wasn't content just to stop with E. coli. He went on to other organisms. We just put yeast and the human up there, and the human, whoops, why did I lose the connection? Because those are the systems that, it's okay. Those are the systems that we are studying in the STC, but there are also other ones like elongatus, a photosynthetic bacteria. You just need to go to the so-called big database that's at his institute, and you can see all the maps that he have. And the reason I like them, even though there are even versions of yeast 8 and 9 being published, you just don't know how well they're curated. And I know what comes off of Bernhard's databases you can trust. So that's, for me, it's very, very important. So now the other programs that were just flashed up for a few minutes. So being able to analyze and calculate the steady state fluxes through all those metabolic networks. He has a program that's called Cobra Pi, which is widely used. Used to be on MATLAB, and now it's on. It's been on Python for years. There's an accompanying visualization program called Escher. I found out just today that he really doesn't even control that anymore. Now it's almost crowdsourced if further developments in it. And that's an extremely important. And now you also see there's some databases that he's developing that's going to help us understand and identify the transcription factors that are really regulating all those cells that he's looked at. So there's one for E. coli, elongatus, yeast, and I think without any further fuss, Bernhard, please start your lecture. We lose the whole connection?
Tell me when you're ready. Tell me when to put it on. So I'm going to talk to you about modulons, which you will know what they are at the end of this talk. I was basically going to talk a little bit about iModulons, which are modularization of the transcriptome. Wanting to be close to a mic, or can you hear me if I walk over here? Okay. And what they're good for. They're good for so many things. It's actually kind of one of these breakthroughs that I've seen in my career. And I took about a third of my slides on that out because I want to talk a little bit about my experience with building genome scale models and whole cell models. And some of that experience might be useful for some of you in the room. So first, modularization. We know that cells are comprised of many, many molecules, thousands of genes, thousands of proteins, maybe even more metabolites. And these are all the molecules that make up a cell, but it's hard to go from all of these molecules to coherent phenotypic functions, which for a bacteria maybe only a dozen or so. They're simple organisms live in simple environments. So the key to understand the omics data types that measure these different molecules or type of molecules on a genome scale, you have to learn how to modularize them. You all see short pathways or molecular assemblies and so forth. But those are small modules, and then larger and larger modules you see in this hierarchy. And the closer we get to a phenotype, these functions actually start to explain the phenotypic function to you. And I'm going to show you today how we discovered these iModulons. So here's a paper from 2019 that started this adventure. And this is Anand Sastry who started doing this. I should say that omics data and the analysis thereof is very democratic. Every gene is treated equally. Every gene has a shot at being in the meaningful output. Of course, those that we know less about don't do as well as the others that we know more about. So Illumina in San Diego, as you know, in the late 2000s, drove the cost of sequencing down to what looks like almost nothing. The first whole genome sequence we did in 2005 of bacteria was $500,000. They dropped that on a log scale by 1,000-fold in just a few years. So right around here was maybe $1,000 or less for a bacterial sequence, and the same thing for RNA. So if you draw this curve here for genomes, it starts shooting up, but transcriptomes didn't start to accumulate until 2013. I'm talking about bacterial transcriptomes, because their mRNA doesn't have a poly-A tail you can grab on, so you have to develop other mRNA purification methods. And once those were in place, this just grew exponentially. And we saw this data coming and coming, and what do we want to do with 100 profiles when you barely know what to do with 2? So this was our early call to arms to build meaningful algorithms to analyze this data on scale. So we're going to see a driver. The drivers for this talk really are cheap omics data of very high quality, and then meaningful machine learning methods that decompose the data into something we call knowledge-enriched output. First paper studied here in 2019, second was Staph aureus a couple of years later, or a year later actually. And these are for Staph and E. coli, because these two organisms, human pathogens that are responsible for almost half the cost of treating bacterial disease. Then we discovered that there's a lot of data, transcriptomic data out at CBI. So we went after species through species. I modularized 13 species, I think, or so, put together iModulon DB. And by late 2020, this had all happened. And that's what I'm saying. This happens so fast these days. Scale of data, scale of analysis, rapid assembly of new discovery and new understanding. And for the last 2 or 3 years, I've had the pleasure of using iModulons for various things. So how do we discover signals in a complex data set? So I found that the best explanation is found on this slide. We all know what an orchestra is. It has maybe 50 source signals. They all come together and produce beautiful music. All the transcription factors or all the transcription regulators all with their signals come together and produce beautiful phenotypes. So roughly the way this works is you spread microphones throughout the room here, and every source signal like this one is heard in every microphone, but at slightly different volume. And the same is true for all the other signals. And what ICA and this class of algorithms can do, they can extract the source signals from a mixed signal that you get. So the instruments of sources here, music is the mixed signal. Here is what we have with cells. We have a bunch of transcriptional regulators. And then we have maybe a very broad condition space. And this is one of the keys, is to get the transcriptome under as wide of conditions as possible. Once you have a good database that is quality controlled, we have R-squared values close to 0.98 to 0.99 for replicates. And so you have a high quality database, you can actually extract these signals. And they are typically between 100 to 150 in a bacterial genome. For E. coli, a little bit better because we have so many conditions. So a little bit of mathematics. So here's what ICA does, this independent component analysis. We take a data matrix, so let's keep that in mind. This is just data under carefully selected conditions. The variables are the genes, they will be the rows in here, and the conditions are the columns, and the number of columns grows and grows and grows. And under a single protocol, We have generated a lot of them, and we have learned that ICA sees through batch effects, so we can often just concatenate a bunch of other data in here, still see the signals and analyze. So this is the histogram of the R-squared values between the two randomly picked profiles. It's right here, and this is our R-squared value distribution for the replicants. So this is a very high-quality database, well-controlled. So ICA takes this data matrix, gives us signaling matrix, where every column in here is a signal. Show you that in a moment. So this is genes by signals. So the signals is what they're calculating. So genes here. And then there is the activity levels of those signals under all these conditions. So that's the same dimension as here. So this matrix is factored into two things with these signals as the modularization that we are seeking in the data set. Here, this is shown in a little more detail. Let's say I take the one column out of the signal matrix. Let's see, okay, you can see the cursor. And what I do here is I'm just doing a histogram of the numerical values in this column. These are the weightings for all the genes in that signal. And this is 0, and this is 1,000 or 2,000. So most of the genes have no weighting. And this is very different than PCA when you get many uninterpretable weightings. But here, the genes in here all have functional annotation, and these are all the genes known to be involved in glycerol metabolism, picking it up, metabolizing it, dumping it into glycolysis. So very clean, very clear signal, very consistent with a lot of knowledge that we have. And then the database here, so across this corresponding row in A, I can see the activities of this signal under all the 278 conditions that were in this original database. And all these tall peaks turn out to be conditions where there's glycerol present. In most of the other ones, this signal is essentially 0. So that's what some iModulon is. It's a group of genes that is coherently regulated across hundreds and even thousands of conditions, algorithmically derived, knowledge-enriched in terms of gene function, and then you can see the strength of the signal over many conditions. And as you can imagine, if there's a white gene, for instance, in an iModulon, this is just a gold mine in terms of conditions that you activate the signal, and you can get that gene to be active. Here's an example. So purine metabolism, PUR is the regulator of purines and pyrimidines in E. coli. So here we have the signal. And you can see in this case, I have positive and negative weightings on the gene. So when these go up, these go down, and vice versa. Most of the weightings are 0. I know the function of these genes, and I can actually here have a graphical visualization of what this cellular function is. And it's a well-known function of purine synthesis. And then a bunch of other genes that are in here too that help, that are associated with this metabolism. Here are just a few select cases from the condition space. The reference is the minimal media, aerobic, 37 degrees. If I add adenine into that media, the signal goes way down. It's basically turned off. Same thing in rich media. If I knock out the regulator, which is a repressor, the signal comes up. If I plot the expression level of the regulator against the iModulon activity, these are now 278 points, looks like this, pretty well correlated. This is not always the case because some transcription factors have a cofactor, like FER, the ferric uptake regulator, has iron as a cofactor as an example. But for many of them, it looks like this. Note, I boxed in a white gene here, so here's a gene of unknown function. But now I see it's somehow involved in purine metabolism, and it is oppositely transcribed relative to the biosynthetic step. So this must have something to do with purine acquisition when you don't have to synthesize it. And looking at the gene, we may hypothesize that this was a transporter, and sure enough it was, and we also found a regulator for that transporter. So once you know this, once you know the cellular function that the white gene is involved with, it's much easier to find this molecular function. Think about this for a moment. So purine is probably one of the best known metabolic functions there is. We all know about this pathway and the function of these molecules. E. coli is probably the best non-human genome that is available. And we didn't know about this transporter for adenine. So there's a lot of missing functions for common cellular functions in organisms like E. coli. And as I'm going to show you, every modulon has at least 1, 2, 3, sometimes 4 Y genes. So this initial study of 278 profiles gave us 92 iModulons. And each iModulon has a story, like the glycerol and the purine iModulon I showed you. Here's a tree map that shows them all. So here is poor R, for instance, 16 genes. Here are some other metabolic functions. Here are some stress functions and alike. So this is a full story of the transcriptome of E. coli and how it behaves onto the conditions that I have. Now I have about 6 times more conditions and I have more iModulons. You can't see the signals unless you have an expression profile under which that signal is active. So these often correspond very well with bottom-up determined regulators or regulons in the literature, which is typically done by some ChIP-exo data of some sort, binding data to the promoter. But the iModulons are top-down, they're calculated, they're inferred from the output of the regulatory network. So these two are different things, but they're often very concordant. Sometimes they're not. If you want to write a lot of papers, you can write a paper on every iModule because each one of them is a meaningful signal. And you can now go into the KO collection, for instance. I could get 60 knockouts of all these genes, treat them all side by side, put some purine stressing conditions on them, and I could confirm the function, the function of those genes in the cellular function. So you can now take these. This is a tree map, gene count regulator or metabolic function in this case, cellular function in this case, as you say, and I can write it out in a map like this. This is what we really like to look at. We love to see this in a textbook or a paper because this is something we can relate to. So these are all the iModulons shown here in various different functions, not just metabolism, but proteostasis. This is in for vibrio nitrogen. So these are the iModulons that we discovered that are involved in competency, which is picking up DNA and incorporating it into the genome. And under each one of these, you see this little pad here, and that highlights the genes in a particular iModulon. Now, what does this look like? To me, this looks like an integrated circuit, an integrated genetic circuit. And here is an integrated electronic circuit as an analogy. And there's a map available to these, a connectivity matrix, which N really is what is analogous to. So this N defines this map. Okay, so it's like having a design to a chip. So we maybe now start to think about the bacterial genome as an integrated genetic circuit. And then I can have the activity level of all of these iModulons under a lot of conditions. And maybe a good analogy is an equalizer that you can use to strengthen or weaken a signal at a particular frequency when you're playing music. So this is kind of the view we are now having or adopting for the functional bacterial genomes. And here's just a little zoom in to show you how many details are in there. Now, iModulons can now be used to study a lot of things. And note that I started with the data matrix. Algorithm that gave me meaningful output, mapped various knowledge onto their gene annotation and so forth. And I discovered something new. I have actually something explained to me in terms of first principles of biology, straight from the data using this algorithm. Now I can start to look at things like the so-called fear-greed trade-off that we see everywhere in biology, all the way from Wall Street, as we've seen in recent days, down to lowly bacteria trying to hedge against risk in the environment. So RPOS is this general stress regulator that measures the stress readiness of the cell, so many different stresses in response to. The growth iModulon here are mostly the ribosomal protein, so the faster the cell is growing, the higher this activity is. So across these rows in here, I have some spectrum like this. I can just plot these two rows against one another to get what we call an activity phase plane. And you see that there is this nice inverse relationship here, where, let's see if I can get this. Here is fast growth, low stress, and vice versa down here. You can look at this color scale here. The high growing cells are here. Slow growing cells are down here. The red dots here are the controls, the reference condition. That should always be in the origin. If it's not, we have some problems. We have discovered in adaptive laboratory evolutions a number of mutations in RNA polymerase that improve both growth and biomass yield. And we can put these mutations simply into the wild type strain, and that moves the point from here to here or here. So this can be used for analyzing causality of mutations with an unprecedented degree of resolution. So we have actually one of the databases we put out is called ALE, Adaptive Laboratory Evolution database, and it has 100,000 or maybe quite a bit more of causal mutations in E. coli now that you could study in this way. Once we have a database like this, well decomposed, you can now start to interpret new data sets. This is done now with a database that has 1,000 samples, a transcriptomic sample from E. coli. Here is a study that we didn't publish, but this is this strain used in another lab, where they took the strain from anaerobic condition, put oxygen in at time 0, 30 seconds, 1 minute, and so forth. So I have a time profile here. I can decompose all of these transcriptomes into iModulons, into the activity level of the iModulons now. And I can generate data like this. This is the ferric uptake regulator, which actually has two-dimensional dynamics. So iModulons are dynamic, regulars are static. And you can see it starts out here, climbs here, overshoots a little bit, and comes down here. Very clear interpretation of this position. And see all these gray dots here in the background, 1,000 conditions. So now the notion of a control changes because the new data can be compared to hundreds and not thousands of other conditions. One of interest, shown of interest here, that's the knockout of the regulator. So up here is the unregulated condition in this faceplate. And I can pick up many other conditions to compare to. Here is the same series of data from blue to whatever the color is, purple, for RK and FNR. So if you know E. coli regulation, these respond to aerobicity. So here is anaerobic, and here is aerobic, and here are a couple other points in the database that represent these conditions, and they follow this trajectory. So now what you can do is you can look at all the iModulons that are changing, and more than 50% of the variation in this data set is associated with 6 iModulons: iron, ROS, aerobicity. You can generate a map like this that might be in a book, and it shows some of these molecular processes here, and this would be the explanation of what happens when E. coli is exposed to oxygen from an anaerobic state. So we have many cases like this now, and you can see iModulons on a genome scale show us how all these signals are changing during a particular transition. And this gives you a first principles explanation of the phenotypic changes you are observing, even tying the iModulons down to individual genes in the iModulons. And even if you have a mutant that you're comparing to, you can see a mutation of a gene in an iModulon also reflected in data like this. So to cap off the mathematics, I want to show this. So here again is my data matrix. Every column is a profile, and then the genes are the rows. This is my M matrix, and the rows here give me the signals. And then the A matrix gives me the activity levels of these signals under a particular condition. So the profile now becomes a linear combination of these iModulon vectors times these weightings that are given in the matrix A. And you don't always fully reconstitute this, but whatever fraction of this you now reconstitute is actually explained. It's not explanation in the sense of PCA when you're fitting in a snug hyperbox around the multidimensional data set. This is explanation in terms of genes, protein, mechanisms, and so forth. So now we can generate this percent explained. So in this case, I had 117 iModulons, and that's this curve. Here's the PCA explanation, but that's it's a statistical measure, whereas the other one is a knowledge-based explanation. We now have a database. I think Sam mentioned this in the introduction. You can go into iModulondb.org and look at the properties of 13 or 14 bacterial transcriptomes. For instance, all of them show the fear-greed ratio. That seems to be a universal property of bacterial transcriptomes, which is maybe perhaps not surprising, except the greed genes are essentially always the same, associated with translation. The fear genes differ from species to species because they are fearful of different things based on the environment they are adapted to. Since this paper in 2019, 40 papers have been generated on the different species, methods, detailed studies that have been done using iModulons. We actually drafted the book of iModulons out of our class notes in the winter quarter. And if you want to see a copy, I'll be happy to send you the link in the public domain. So I don't see a clock in here anywhere, but let's move on. So what are iModulons good for? We can scan through this list. They're good to do all of these things. This is often give talks about biomanufacturing or biosustainability applications. I kind of tend to focus on these first four, but I decided to drop some of them so we could talk more about genome scale models at the end. We can deorphanize, like I mentioned. We discovered a number of DNA transfer processes in terms of iModulons. Zan also mentioned Synechococcus elongatus. We found all the iModulons in there, even the carbon concentrating mechanism which has 2 iModulons. And now we know that there are about 8 to 10 Y genes with every one of these modulons. There's a lot of gene function to be discovered in this organism. And biosynthetic gene clusters turn out to be iModulons if you're looking at places like Streptomyces, and we can actually show we can learn the regulation of them. And this is a $350 billion industry a year, produce these secondary metabolites. The most dramatic application here is cross-species transfer of iModulons. And this was kind of a shocking result, actually, how well this worked. So the hypothesis is that each one of these signals is independent. All these genes that are in an iModulon are a cellular function. So we said, why don't we take a cellular function from one bacteria and put it into another and see if it boots up that function? So that's what cross-species transfer means. So there are a few traits of interest. So this one shows that there is an iModulon in Pseudomonas aeruginosa that responds to penicillin-like antibiotics. So here's a series of them. This is the activity of the iModulon. Here are some other antibiotics not responsive to them. So this has something to do with penicillin resistance or penicillin response. Genes, this is a location of the genes on the chromosome, so 6 million base pairs. See the genes are spread throughout, and only one of them really has a solid functional annotation, and that's a beta-lactamase, which is not unexpected in this case. There's a bunch of other genes in there of unknown function. So then we refactor, as shown there on the gene map, these genes, put them on a plasmid, and we also made another plasmid with just the beta-lactamase. So here is titration of inhibition with ampicillin. This is E. coli by itself, easily killed. Here is E. coli with this full iModulon, becomes very resistant. This is a log scale. If you only do the beta-lactamase, you get something, you get this curve. So this is about 4-fold different. And this is the donor strain. So we are doing this now in mass. We're taking all the major human bacterial pathogens, exposing them to all the major antibiotics. And we have a database now called Antibiotica, ICA, always have ICA. This is basically going to be a map of AMR mechanisms, antimicrobial resistance mechanisms. Here is another application of iModulon transfer. So another Pseudomonas species called Pseudomonas putida can metabolize 2,3-butanediol. Most other bacterial strains cannot do that, including E. coli. So here's the pathway. And here, like I showed you the Vibrio map, every catabolic pathway shows up with their own iModulon. That happens also in Putida for this substrate, that they're all here. So they're basically co-located in roughly an operon. Here's the pathway. These genes underlined in blue are genes for the enzymes known to be in this pathway, but there's one Y gene in here, which is this one. So we constructed a few plasmids to move this trait into E. coli. One is the full iModulon, and one was just a knowledge-based pathway, then an empty plasmid. So here's the empty plasmid. These blue bars here are the concentration of the substrate. This is the concentration of this intermediate. Empty plasmid, so E. coli can carry out this reaction here to a certain extent, but can't complete the pathway. Here is the pathway as moved from Putida, just the pathway genes. And I get this profile here, so more is consumed, but it's not fully consumed. The substrate is basically either in this metabolite or unconsumed. If I include the Y gene in here, process is complete. So this Y gene has to be there to complete this pathway, presumably associated with something down here. So this is the cross-species transfer of a catabolic property that works if the Y genes are included. Basically confirms the fact that that Y gene, even if it's of unknown molecular function, is of known cellular function, and we can execute those pathways. The second application I want to talk about is media design. Now, which media do you use for your organism, and why? Nobody knows. These things come from a screen from the 60s or 70s, and they have just used for that species forever, right? For E. coli, it's M9. Most commonly used media. Yes, people like to use it. Well, I don't know. So I did a book. Everybody uses it. So I'm going to show you next is that we can now design media based on first principles. And that's simply by looking at the activity level of the iModulons in the media you're screening, all the conditions. So let me show you how that goes. So here's one example of showing you how surgical this is. Here again is the summation of the iModulon activities. So here's a comparison between two conditions, the reference condition and the reference condition plus one amino acid methionine. And the 45-degree line shows that almost all the iModulons are exactly the same activity under the two conditions. The only activity that has changed is here, which is the methionine iModulon, which is downregulated because it's no longer needed, and LRP. So this is the leucine responsive protein that's kind of a master regulator associated with amino acids in E. coli. How about that? I can analyze it with one substrate addition, change the substrate. I can really find out what the best media is for an organism. So here is such a screen for Vibrio nitrogens. You see a bunch of substrates down there. And here's an activity level of a bunch of catabolic iModulons. And this is kind of, so every column in here is the activity level of all these iModulons under a given condition. So it's like one setting on this equalizer. And you can see the boxes in green, they kind of match. There's ramenose iModulon. So there's kind of a diagonal line that goes across here that makes perfect sense. But there's a lot of off-diagonal elements, if you will. And these two iModulons that showed up were of completely unknown function. And you see that they are activated for some of these substrates, but not all. And this turns out to be a pleiotropic transporter system. I can import many different compounds. So here's a bona fide discovery of a new cellular process and functional annotation to those genes. I can do the same thing with stresses. So here I have a bunch of stress iModulons, misfolding, ROS, osmolarity, membrane stress, and so forth. CSP, cold shock protein response, shown there. So the phages are activated, prophages in the genome. Bunch of substrates again. These are green, so they don't do much. So you see the color bar over there is high. Red is upregulated, blue is downregulated. So these are mostly pale colors here. But here's a bunch of substrates with red on them because they elicit one stress or another. Some of them actually just, these three here elicit a misfolding stress. Now, why is that? Ethanol is in here, it activates the membrane stress that is well known. So you can really treat that as positive control. And then we have the rich media, that's supposed to be so nice and gentle and treat cells well. Well, here is the same thing for rich media. Some of these rich media formulations are not benign at all when it comes to stresses inflicted on the host. We have now taken this to scale with E. coli. Many people are familiar with CRP here as the master kind of repressor, and certain substrates are in there. And we've now done a big substrate screen like is shown here. And here are some iModulons rank ordered here. And this is the data coming out of the ICA. You analyze this, and this actually falls into 6 different classes of iModulons and 4 different groups of substrates. And over here is an explanation as to what all of these substrates do. And we are now doing FDA calculations to figure out ATP efficiency, thermodynamic efficiency, and things like that. And if you sit and look at this a little more closely, you get a diagram that looks like this. And this is a full expose of the metabolic or carbon substrate selection criteria that E. coli uses. And so now we're doing dioxys, taking it from one substrate to the next, and see if these iModulons come up and down based on how we have predicted, how this predicts it. We're now also doing organic nitrogens. That's the second batch that's coming up. It has a very similar diagram as this one. And then we have a large number of single nutrient perturbation experiments, like I showed the methionine before. And so all of this is data that can be used to design a media that is as stress-free as possible. And it's not just stresses, there are a whole bunch of other processes in the host that can be activated by the media. This I'm going to skip, but I would say the same thing is true for heterologous gene expression. Many of you are probably expressing heterologous genes in E. coli. Every gene expresses differently in E. coli. And then I do a screen of about 60 protein, and I look at the activity levels of the stress iModulons. I see that every protein activates different stresses in the host. Some very strong, and then they become very poor expressors and producers of that protein. But these are probably serendipitous responses because E. coli has never been impregnated by this gene and has a bunch of this protein floating around, so it probably just activates some stresses. We know how to mitigate all these stresses, as I showed you in the fear-greed ratio plot. And so now you can put mutations into your production host and mitigate the responses. So this is really interesting. We have also continued to hack into genomes, if you will. Extracting iModulons, we see hacking into a genome. I'm hacking and I get the process that I'm looking for. And here are some examples of cellular processes we have hacked out of a genome. The antibiotic responses, the biosynthetic gene clusters for secondary metabolites, phage infections, competency I mentioned. This will probably end up as being a synthetic horizontal gene transfer systems. I think we pretty much now have the electrosynthetic iModulons, and I mentioned some of these other ones. Awareness is kind of interesting. We've decided to activate all the two-component systems in E. coli, of which there are 27. I think we have functions of 23 of them. And when you activate them, you really pull the transcriptome apart. It's really, this is the awareness of the organism, which is probably what we found biologically we are maybe the most interested in. So to finish this segment in the talk, as I said in the beginning, we're in a new era now where we can generate in mass high-quality omics profiles that are reproducible and protocols transferable from one lab to the next. And then we can develop novel algorithm and workflows to extract information, biological information out of the data, and that becomes understandable in terms of first principles. This here is a series of different analytics we are developing. I showed you this one. We have similar results for pangenomics. So we're comparing tens of thousands of genomes of strains of the same species. We just put this out live where anyone can get the full structural proteome for a bacterial genome that you're working with. And that's, everybody knows the magic of AlphaFold, and you need some additional capabilities to drive that workflow to completion. And so we can now be doing that, and then we can map all the mutations, for instance, onto the structures in mass. So a lot of discovery of causal mutations. Zan mentioned this. We now also have, we also socialize genome scale models that I mentioned these two. So going forward, integrating these three together is going to give us data-driven explanation of how these bacterial cells work. And we are now filling out matrices like these. So every column in here is one of these data types I showed you before, databases at the end, and all the organisms we've been working with. This is our slide for industrially relevant organisms. We have a similar slide for human pathogens, and then the software has been available. So there, as Stan said, we put effort into socializing what we do through good software. So if you want to publish a lot of papers, you can pick an organism, just apply this one thing after another, write one paper at a time, maybe in 6 months or so, depending on how quickly you generate the data. And then the issue becomes interoperability. How are we going to make this interoperable? How am I doing on time? There's a clock over the...
It's 2:40.
Oh, okay. So I got like 50 minutes. 20
Yeah.
minutes. So that's what's happening with big data analytics. This is not AI. I'm very pessimistic on a revolutionary results from AI for many of these data types. Structures, obviously, AlphaFold works, but it's not for all of them. And that's because we just don't have enough data to learn AI. So I think these machine learning methods and algorithms, knowledge-enriched, they kind of will give us what we want, at least for bacteria. So let's talk about genome scale models and the whole cell models. The former are kind of driven by genomics and genetic parameters. The latter are more biophysical in nature, and the two shall meet, as we've seen. And quite frankly, this is going to be one of the basic ingredients in a new field that is coming up. And I think we will see departments of genome engineering before too long. Maybe genome science and engineering, like computer science and engineering. But they will naturally fall into different colleges depending on what university you're at. I was actually in Korea last week, and KAIST has actually built the first department of this type and has 20 positions open. Anybody's interested in going to Korea, designing genomes. So in terms of analytics, the way we teach analytics of biological data is now around these 4 levels. The 1 and 4 are familiar to you. So there is multivariate statistics that we are all familiar with, PCA, plots, cluster maps, etc. But this is data analysis. Here is an example of what I showed. The iModulons are in here. They are knowledge-enriched data analysis to bring it actually down to biological variables we like. But this is still data analysis. I mean, I just have a group of genes, they're activated or not activated. But to know how all the iModulons dance together on a genome scale, we're going to need models, computational models, that take everything into account at the same time and can actually calculate the homeostatic state. Most people who do data analytics don't appreciate what this means. You have to be a biologist to fully understand homeostasis and evolution, how important this is. Being able to calculate or predict the homeostatic states is just so important. So this is, of course, the genome scale models scale. Then if you go over here, these are the models that are driven by physical or biophysical principles, a fermenter in action there or a flagella. And now you want to do this on the genome scale and are faced with the usual parameter estimation challenges and whatnot. But these here will meet in time. Maybe they already have. So these are the levels of analysis. And if you have departments of genome engineering, you'll have all of these things active in such a department. So I, very, very early in my career, as a senior graduate student, and then as an early faculty member, I got fascinated by enzymes and enzyme kinetics. And at the time, the mantra was that you can just component by component build up models of cells by knowing the enzyme kinetics, binding constants, and so forth. And of course, now we all know that that's a very, very difficult challenge. But shockingly, we were able to take a lot of data that came out of former East Germany and build a whole cell model that actually computed the physiological states of the red blood cell. And you could simulate it, and it reflected a number of the properties of the cell. Then a few years later, we put almost data in here. I took 24 students in on a Sunday morning to the lab in a fasting state, hopefully not a hungover state, but in a fasting state. And I had an MD in the lab that took blood samples. We did the genotype, we did the exometabolome and the endometabolome for the red cell, and we built personalized red cell models that turned out to be predictive and explanatory. So we call that the 3rd generation, and we are about to put out the 4th generation model. So for the human red blood cell, this is possible. You can get the biophysical model here on scale, and there are some large NIH programs been run lately, REDS3 most notably, they generated an incredible amount of metabolic data, proteomic data, and genotypic data for donors of red cells. So that's just a wealth of information to build a fully 3-dimensional biomechanical, full model of this red cell. The red cells are not uninteresting or unimportant. 85% of your cells in your body are red cells, as an example. And I got long COVID last summer, and my oxygen saturation dropped below 0.9 to 0.85, and I was almost fainting. So my red cells were just not loading because my lung capacity was so low. just sitting up and typing an email was exhausting. So red cells are pretty important. You know, you don't want them to fail. They are also targets of synthetic biology now in design, as we saw with correction of the mutation that causes sickle cell anemia in the CD34 positive cells. And it would be nice to have a model of a red cell and the differentiation process that generates them from stem cell. So there is some substantial difference between how people think about modeling in biophysics and in engineering. In engineering, we are taught these universal laws that always apply. Navier-Stokes equation would describe all fluid mechanics. It's fundamental. You just have to adopt it. Same thing with molecular diffusion. Books, long classes taken on the very kind of biophysics center. So that's how people think here. But with genomes, so genomes are relatively new. They're around for about, well, actually 3 decades now, so maybe not that new. But you still don't know what's on here. We don't have equations like this for biology yet. But we can take at least the genes whose function we know and build these matrices like this one here. So you can maybe these 2 genes form 2 proteins form a heterodimer. And then I can build a matrix like this. Where the rows are the components, and here are these 2 proteins and form a heterodimer. So I can write this out like a reaction. And here is the transcription factor that I can bind to a promoter. I can write that relationship out like this. And if this dimer is 4, maybe it catalyzes the reaction of A to B, and I show that over here. So this is like a 2-dimensional genome annotation. It's not just what the genes are, but how they interrelate. And this is the basis for the genome scale models mentioned in the introduction. And their issue is such that they don't give you a unique solution. As you know, there are often alternate solutions in biology, and organisms evolve. The genome can express different phenotypes. So there is some malleability inherent in these models, these genome scale models, and they're very simple. It just takes every metabolite, every compound of interest, identifies all the reactions that use to degrade that compound. You write the simple dynamic mass balance. You put it into a steady state because metabolism tends to be relatively rapid. You have the simple flux balance equation, and that's it. And you solve it by linear optimization. Very simple mathematics, very simple calculation. And I ended up writing a 600-page book on this, which I will never do again. It took like forever to do. And there's a nice review paper on them here. They are interesting and kind of biologically meaningful because they explicitly account for genetic parameters as described there. And here's just one example of that. So a reaction like this PGI reaction here, glycolysis, is linked to a gene. It's linked to a transcript, protein, and the gene. So if I knock out this gene, this goes away. And that's just described by a Boolean statement. No on this, no on this, right? And this, of course, there are much more complicated assembly reactions of protein complexes than shown here. So you can actually apply this to pretty complicated stuff. The possible solutions called the solution space is a polytope, like shown there, that can be further shrunk if you have regulatory and kinetic information. But you can also describe evolution or adaptation of cells to a different environment using these models. And this is why biologists liked them a lot. They were adopted in the early 2000s almost right away. And Jared was a PhD student in biology and came to work in our lab to use them. And he says these models represent how biologists think about biology. They think about constraints. What's the limiting nutrient? What's the temperature? You know, they always think about constraints under which biological organisms operate. And then there is the interest in the internal mechanisms. How do they mitigate ROS stress, etc. And we can now do this on a genome scale because we have the genome sequences and we can annotate them and we can actually get this on a genome scale. And then inherent in biology is a notion of optimality, adaptation, evolution, and all sorts of things. And when Jared pointed this out to me, I made this slide. Because so these models represent this. And so when biologists ask questions, they can very often be answered by these simple models. And here are early examples of this. Here's the basic procedure, genome reconstruction, solution space, picking a solution. And then we can apply this very simple formalism. So here, for instance, is oxygen octigrade, substrate malate, in this case octigrade, and this is growth rate. This is a three-dimensional projection of that high-dimensional polytope, and you make predictions of where the best growth rates are. They're here. So this is a lab strain, been used for decades, and probably seen malate quite often along the way. So it's basically satisfy that optimality principle. You try other substrates like glycerol, that's not the case. So we're gonna annoy. We couldn't predict this because we predict this for so many substrates. That's what led us to do a laboratory evolution. We just figured the strain hasn't seen enough glycerol. It doesn't know how to do this optimally. And we did the laboratory evolution. And sure enough, these are evolutionary trajectories that end up in the predicted spot. So then we challenged them even further. We took a bunch of knockout strains, measured the growth rates, the blue dots prior to evolution. Then we evolved them, got the pink dots, and they're all pretty much on the 45-degree lines where we were comparing experiment to computation. And this was very shocking that this worked so well for such a simple model. This got scaled up massively when knockout collections became available. You could go all of the knockouts under a lot of different conditions. So you could start to look at hundreds of thousands of conditions. And this is probably the largest attempt at computationally predicted phenotypes. That's why science took this commentary or perspective. And you can see the organisms for which this data was available here. And for the single knockout libraries, we were getting over 90 to 100% prediction of essential genes under a given condition. And this was kind of shocking, but it basically tells us that the missing equation I had on that mass transfer slide is almost complete. We are missing something, there are some errors in it, but it is largely there. And then we can do more sophisticated stuff like this. There is now called GWAS, something called GWAS FBA, and not getting into too much detail here, but we were studying antibiotics, and we had sensitive and resistant strains, and they could be located in a flux space that's shown here. And then we use support vector machines, SVMs, to find the best plane that separated the 2 points. And that's shown here. And since this is a flux space, this corresponds to a metabolic process. This plane corresponds to a metabolic process. And that metabolic process is shown to the right in this slide. And all the known resistance genes associated with this antibiotic in this strain are marked there with these check marks in green. So FDA can give you that whole pathway as being the pathway that separates resistant from sensitive strains. So FBA can be used for all kinds of interesting purposes, and it's a relatively simple model. Then we built the so-called me models that had just metabolism plus expression. And today I've been dazzled with the details of this at my different meetings, but this is just straightforward stoichiometric representation of this in here, not in space or protein complexes, but just the overall process. So now we have a me model that is metabolism, gives all the precursors for expression, translation, transcription and translation, and makes the proteome that this model here calls for. And we will also have all the ions and the other cofactors embedded in them. In addition, we have built the proteostatic mechanisms, the temperature mostly, ROS responses, and pH responses. And the first one of these models, called fold-me, because it had this folding or proteostatic system in it, I should say, calculated this curve. The black curve in there is calculated. And this is a temperature sensitivity of E. coli growth, just calculated. And the data are different data sets that have been published. I was just blown away when I saw this result. And what happens here post-peak is that the proteome starts to unfold on this fold due to temperature, and that little module down there becomes active, and the proteome allocation to chaperones and other protein in this proteostatic mechanism becomes very large. It takes all the protein away from the growth functions, and that's why this goes down. So proteome allocation is just such an important principle in the function of cells, as I'll maybe say more about. So let's see, what else do I have here? Um, uh, uh, so me models are basically OpEx and CapEx. So OpEx is just the flux matter where it comes to run the cell. CapEx is the investment in the proteome to keep it wholly going. So these are very econometric models and econometric thinking about this. So proteome allocation is also turning out to be an organizing principle of, or not turning out, looks like it's an organizing principle of communities also. So finally, you're all familiar with these images. You have stepped up here to be so brave to take these genome scale representations and convert them into biophysical models. And so you're going through all the challenges of representing this in space, in 3D, and in time, with accurate kinetic constants. They're making a lot of progress. I could have never dreamt when I started this process that this was possible one day. So congratulations. This is just fantastic. I just want to end with this slide. I think this is my last slide. To emphasize that when we build these models, we need to understand that there are universal physical chemical constraints on all cells and all cellular processes. And these are, we call these abiotic constraints. Since they are so fundamental, to call them the ABCs. Every model we build has to satisfy these. Charge balance, osmotic balance, pH, thermodynamics, and all these things. And we've actually used these models to do two things. We have built under these constraints a reconstruction of the postulated pre-abiotic TCA cycle, shown here. Most of this chemistry is shown in one lab or another. And we can now put this under the constraints of these thermal hydrothermal vents, I guess they're called, at the bottom of the ocean, and they can solve. So this postulated system actually can work under these constraints and under these conditions, but the solution space is very small. And it has two interesting characteristics. Every point in it has growth because the osmotic pressure goes up as it sucks in water. So growth may have been a part of life from the very, very beginning. So that's an emergent property that comes out of this model. And the other is the solution space is closed in some dimensions, like the electrochemical gradients that are created, but is open in the direction of K-cats. That's, of course, where evolution goes straight up. So if this happened in the beginning, evolution had to go up in that solution space. So we are now putting actual real CO2 fixation processes in there. So we put the Langdali pathway in there, and we put the glycine cleavage pathway in there. And they have just a minor stoichiometric difference. But when you assess their thermodynamic efficiency by doing this in output, one is much better than the other, even if it's just a minor change. So I remember these fundamental constraints from my first whole cell model of the red cell. And I realized pretty early that these have to be satisfied if you want to get reasonable solutions. Okay, so actually I'm going to finish at some point, and that would be now. So I will say first that sequence-based data types are affordable and available on scale. I have kind of given up on metabolomics. It's too difficult, too expensive, too much variation, batch variations. Best of luck to those that want to generate those data sets. And the center I ran in Denmark, I spent $15 million on mass specs. I put 6 FTEs in there, and I never got a useful data set. Big data set I'm talking about. Of course, it was useful for so drilled down experiments. But we now have large and growing DNA-seq and RNA-seq databases available. They're not going to shrink, they just keep on growing. And we've seen with every decade that's increase in size of these databases, we learn something new that we didn't know about. For instance, at 10 to the 3, we get the allelome. So we get thousands of alleles of the same gene. Let's take a favorite gene like PFK. Or any other gene across all E. coli strains or all staph strains, we can find exactly the sequence variation of this in the whole species in that gene. And it's tiny, which is very good news for modelers because kinetics are not changing. I have a slide that asked me a question that shows how I support that statement. We can actually integrate with some algorithms that can see through, so we can integrate data from different labs using algorithms that can see through the batch effect. A lot of novel data analytics needed. I'll bet you that just like with the Google search algorithm, the ICA algorithm is going to continue to evolve and get better. I'm going to learn more from it. And the ICA of the transcriptomic data is extremely useful. It's actually shocking to me that how predictive it is and how it describes biology accurately. I'm sure for some things it's going to fail, but so far we have not found those failures. So knowledge enrichment is very important to large databases, like I mentioned. And finally, my examples were focused on this, show the utility in synthetic biology and industrial biotech. So with that, I'm actually going to stop. And I've been funded by the NIH, DOE, and very generously by the Novo Nordisk Foundation in recent years. I'll take any questions from anyone.
Yeah, you covered a lot of ground, right? Any questions? Yeah, I got it.
Really great talk. The way to analyze the transcriptomes using iModulons is really interesting, and especially when you show all these bacteria and you pull out gene clusters, which makes so much sense, right? What I'm curious about really is, I think that the implications of this on eukaryotic transcriptome data set are quite enormous because they're not clustered.
So we started doing this for yeast data sets, Pichia, Saccharomyces, and so forth. Surprisingly, there's not this data available for them, as you might think. And the iModulons are there. The signals are there. But what's different about eukaryotes is that the regulation of these genes is very complicated. And there are a lot of different regulators involved. We don't get these clean iModulons like we saw iModulon and so forth. And the other thing, surprisingly, especially with the human data that we looked at, its quality isn't good enough. I don't know. It's probably because the mRNA is in many organelles, and it's difficult to get the reproducible profiles. They're much more difficult than with bacteria. So we've done some calculations there. So we'll see how that goes. But I think it's a little early. And the best way to do this is when you have a time-dependent process. We like that wound healing samples. That's why I think differentiation into erythropoiesis, which is 10 days, I think you will find all the iModulons associated with erythropoiesis if you have a good data set there.
Yeah, same with adaptive immune system, I think.
Adaptive immune system, yes, exactly. Right.
Vito, you had a...
Yes, so following on that, but back to the bacteria, as you said, this is the first approximation to the activity of every gene.
Yeah.
Can you give us a sense how good is that approximation? How should I think of the next term for a typical gene? How much of it is inexplicable by the single main variable it lives in, model in this case?
So I think the condition space is the key to that question, answering that question. So we're always growing the bacteria minimum media in the lab. We change some variables. So maybe that's one of the reasons why there's a dominant regulator for each one of these iModulons. If you take it into very unusual circumstances, maybe there's going to be another regulator that gets involved then. And then we would get what's called bilinear effect in the summation of these columns. It would be more than one factor affecting one signal. The signals, I don't think will change that much, but I could be wrong. Sometimes we do see fractionation of a signal into two subsignals when I add new data into the database. But it does seem to a first approximation, it is really good, actually surprisingly good. But I think we're all aware of the fact that there is probably going to be nonlinear, maybe bilinear effects discovered in these networks eventually, and maybe there will be a bilinear ICA algorithms available in the future. But we're very happy at the moment.
Yeah, I remember when Yuri Alon was doing his pioneering work back in 2000, trying to look at the key, regulators in E. coli, and he had sort of a list, and now that list has just grown. So it'd almost be interesting to go back and run it just on the conditions that he was able to get at. I think that's the other thing we don't appreciate, that you've generated, or one can generate so many more conditions, and then can do the omics studies on it that you would need to feed it into this. So I thought his work was fascinating.
Yeah.
Uh,
So,
So it's really, any further questions? Because I know people are waiting to talk with him afterwards. So maybe we thank you one more time